Hello everyone. What we're going to talk about in this second biochemistry lecture is all the processes that lead to the synthesis of new nucleic acid. Therefore, what we'll talk about here is the process known as DNA replication, the process where DNA is copied into an identical DNA molecule. Then we'll talk about the process of transcription, that process where instead RNA, so a new nucleic acid, new type, so ribonucleic acid, is produced by copying the DNA with the deoxyribonucleic acid. And then we'll talk about also some different ways in which nucleic acid can be produced, methods or processes which you may be less familiar with and which take place in RNA viruses, those viruses that do have an RNA genome, not a DNA genome, where you have that new RNA is made directly from RNA through the RNA-dependent RNA replication, or is first converted into DNA and then back to RNA through what is called reverse transcription. So starting with possible models of DNA replication. First of all, what are we talking about here when we talk about DNA replication? What we're talking about here is that process that takes place during the cell cycle. The cell cycle is the series of events, if you recall from Bio 2, whereby you have that one cell goes through all this cycle of events producing two identical cells, and then can go back to the same events dividing again into two, and so on. This is what we refer to. Now, this cell cycle is different in prokaryote, as you can see here on the left side, than in eukaryote here on the left side compared to the prokaryote, where it's simpler in the prokaryote. The reason being that prokaryotes have only one chromosome to deal with, and they don't have a nuclear member to deal with. And so, for this reason, the eukaryotic cell cycle has also a dedicated mitotic phase. Anyways, the process we're talking about here, DNA replication, is that process that takes place during the cell cycle, during interphase in eukaryotes. That's where the DNA gets duplicated into two identical copies, or what is called a phase B and C here, which are overlapping. Theoretically, it will be phase B where it's duplicated DNA, and C where it gets separated in prokaryotes here. So that's the process through which the DNA molecule is copied into two full identical copies. Now, we know, and you know already from Bio 2, we know it because we hear it so many times, that the replication of DNA is referred to as a semi-conservative process or semi-conservative process. What does it mean? It means that now, being formed by two opposite, two antiparallel strands DNA, you have that the DNA opens up, each strand acts as a template to produce a new template, a new strand, the antiparallel strand, opposite, complementary to the original strand. And by doing that, you end up having that from one original parental DNA molecule, you start having two molecules which both have one parental strand and a completely new antiparallel strand attached to it. So each molecule would have conserved half of the original DNA in the form of one of the two strands, and therefore is referred to as semi-conservative. But how did we get to know that? This was, by the way, a model that was already proposed by the same scientists, Watson and Crick, that had discovered the structure of DNA. And in fact, their structure is based, in fact, on the knowledge of complementarity. The fact that there are two complementary strands based on the Chargas rule, right, allows you to predict from one strand the sequence of the other one. And so they immediately suggest, even though they didn't have any evidence at that time, that it will make sense if one strand could be used by some sort of enzyme, we know now is DNA polymerase, to form the second one by using that complementarity rule, by adding one nucleotide at the time. So that's exactly what they had predicted, but they didn't have any evidence at that time. How did we get to know that indeed DNA is replicated semi-conservatively? Is thanks to the experiment done by these two scientists, known his name are Meselson and Stahl, where basically what they did an experiment in bacteria, and they follow what happens to the DNA when it replicates for 1 or 2 generations. We'll talk about the possible model in the next slide. But how did they follow how new DNA is formed? Is by using a radioisotope of nitrogen. You know, nitrogen is a key component in the nitrogenous bases of nucleic acid. And so in this case, DNA, right? It's present in every single nitrogenous bases. That's the reason why they're called nitrogenous bases. And now, bacteria or living organisms, they don't know any difference between different isotopes of nitrogen. So there are different isotopes of nitrogen, 14, 15, and so on. For them, it's the same thing. They would integrate any isotope into these nitrogenous bases when they synthesize them. So if you grow them with one isotope, as Meselson did, now you would end up having that. After a few generations, all the bacteria would have that specific isotope, if it's the only one that is present in the media, in the growth media. So that's what they did. They grew their bacteria, and they make sure that all DNA would contain N15, the isotope 15 of nitrogen. Then, to follow what happened, so this would have been referred to as the parental DNA. And now, to follow what happened after some generation, they switched this bacteria from a medium containing N15 to a medium containing another isotope of nitrogen 14. At this point, now the bacteria, if you switch them to nitrogen 14 containing media, they will integrate N14 in the newly synthesized DNA, right? So after 1 generation, they will still have some nitrogen 15 DNA, but the new DNA will be containing nitrogen 14. How can you distinguish how much of N14 and 15 DNA you have is due to the fact that the difference that there is between these isotopes, there is a difference of 1 neutron between them. Only 1 neutron. It sounds very laughable as a difference, but if you multiply that by the billion of nitrogenous bases that all the DNA combined together would have in this bacteria, now you start having a significant difference in weight between the new DNA containing N14 and the old one containing N15. How can you visualize that? You can wait for years, leaving a sample there and waiting that the DNA would separate based on their weight. But you can make it much faster through centrifugation. So you can take any DNA containing N15 or N14 or a mix of them, put them in a test tube, and put at a very, very high speed in a centrifuge. There will be also a gradient inside here. You're talking about 50,000 RPM, rotation per minute, super fast. By doing that, basically what you're doing, you're accelerating gravity, right? Instead of waiting with a sample there on your bench for years and years until you die and then you don't have a result. In this case, in a matter of 1-2 hours, you can actually speed up this process so much. You get thousands of Gs in this sample. And then the samples, based on what type of isotope would have, will go faster down in the test tube compared to those that have the lightest form of the isotope. So, for example here, now in these bacteria here that have been grown in N15, you will know that all the DNA will contain, both strands will contain N15, and therefore will be a much heavier DNA compared to the one containing N14. And so, in fact, if you centrifuge, it will give you a band, a visible band, you can actually see it, that is closer to the bottom of the test tube. Now, if you start having a hybrid one, so imagine this, you do 1 generation and then it's mixed now based on the semi-conservative model. So you would have one strand with N15, the other one with N14, you will get something that is lighter than the pure N15 DNA. So you will get about this higher. Now, if your DNA becomes pure with N14 only, at this point it's even lighter. It will give you a band even farther from the bottom of the test tube. Great. By doing this, now Meselson and Stahl had a way to basically follow what happened to the DNA. Just by changing the type of isotope and centrifuging bacteria, they could actually visualize the type of DNA that these bacteria would have after 1 or 2 generations. Now, one step back. What are the three possible models of replication that the DNA could use to create two identical copies? Well, we know the real one is the semi-conservative replication method, which is the one where you have that the parental DNA molecule has the two antiparallel strands, and each strand is used to produce a new complementary strand which stays attached to the parental strand. And therefore you end up having two hybrid molecules, each one of them having half of the DNA from the parent cells all in one strand and one new strand attached to it. And they will stay like that, and these obviously will go on for the next generation. So if you keep going, now you start having more and more DNA that is completely new compared to the first generation or the starting generation of DNA. However, this does not exclude other models. Like the fact that this makes sense, right? And that's what Watson and Crick were talking about. It makes sense. It doesn't prove anything in science. You have to exclude any other possibility. What are the other possibilities? Well, one is that DNA could be replicated in a conservative way. Overall, it's similar to what a semi-conservative replication method would be. So each strand works as a template, but then the new template somehow, the new strands, somehow bind to each other, whereas the old ones somehow go back to bind to each other. And therefore, you end up having a new molecule here in red, completely new. Both strands will be new, and the other one will be old. And this could go on for several generations, right? That's referred to as a conservative replication. So because the original DNA is conserved after replication, the full double helix is conserved there. A third possible model is what's referred to as the dispersive replication model, where you have that the parental DNA molecule is copied in some parts. One strand makes the new part, and the other strand makes the other part, and so on. And then these mixed stay in the two molecules that are being generated. You can see how basically it's very not intuitive, but again, cannot be excluded this method. And what you end up having here is new molecules here with both strands having some of the old DNA and some of the new DNA dispersed among each other. That's why it's called dispersive replication model. And then with time, obviously, more and more of this new DNA, of the DNA found in the next generation, will become more and more of the new type compared to the original one. Okay, so these are the three possible models: semi-conservative replication, conservative replication, and dispersive replication. How did Mesenthal and Stahl excluded two of these models, showing that only the semi-conservative replication model could be the one possible? Well, they did it as we said before. So they started with bacteria that were grown only in N15, the heavy form of nitrogen, right? So all their nitrogenous bases would have contained N15. So if you had taken the DNA here and centrifuged, you would see only this band right here, not the other two, right? That's what you would expect. So starting from there, then, what they did, they switched. They switched the media, so they took the bacteria, they separate them, they remove the N15 completely, and they put them in a media containing N14. So what it means that now, if you wait for one generation, it means that the new DNA synthesized will contain at this point not N15, but N14, the newly synthesized DNA. Obviously, you will still have some carried over N15 labeled DNA from there. Now, what would you expect to see here? Now, if DNA is replicating in a semi-conservative way, you would expect to have, again, as we said before, one strand completely coming from the parental DNA and one completely new. Therefore, this DNA, the double helix here, will be a hybrid of both. It will be a mix of N14 and N15. Practically speaking, what it means is that if you take these bacteria and you centrifuge them, you would expect to get only one DNA band here in the middle, this band right here. Now, if instead DNA was replicating in a conservative way, what you would expect to get at this point, you would expect to get that some bacteria would have the original DNA and some other would have a completely new DNA. So these would have only N15, the old one, and this only N14. So what you would expect to get now, if you take this bacteria, extract the DNA, and centrifuge it, you will get the two extreme bands. You should see on the test tube this band with very light DNA corresponding to the newly synthesized DNA, and this band here with very heavy DNA corresponding to the original DNA still present there. Now, when it comes to the dispersive replication model, instead what you would expect to get is, as we said before, some parts will be coming from one strand, some or the other one in terms of new DNAs. You will get that both strands now in the newly generated cells will contain a little bit of old, a little bit of new DNA. And therefore, all the double helix here will be a mix of N14 and N15. Same thing for here and for the other one. And so what you would expect to get is a band roughly in the middle right here. What did they see? Well, they took the DNA and what they saw is this. They indeed see only a middle band here, a band that would correspond to a hybrid DNA, something that contains both N15 and N14. Therefore, these data that they observe support only what you would expect from the semi-conservative replication and then dispersive replication. By doing that, they excluded the conservative replication. Still, this does not exclude the dispersive or the semi-conservative replication model. How did they exclude that? Well, they waited for a next generation. So they let them go again and they did the same thing. What would you expect to get here? If DNA is replicating in a semi-conservative way, that's what you would expect to get at this point. Now, these two strands separate, each one act as a template and stay combined with the newly generated molecule. So if the old template here, the N15 strand here, gets copied into the new one, this would be a hybrid of N14 and N15. But now, these newly synthesized here in generation 1, or after first replication, I would say generation 2 here, probably, now gets copied into a new one. Now you will start having some pure N14 DNA molecule appearing, right? So when you centrifuge, then at this point you would expect to get two bands, one with an intermediate weight and one with a very light weight. But you would not expect to get an N15 band. So only these two bands should be present there. Now, what about the dispersive replication model? Well, in that case, now you would have that again. Each strand gets partly copied, and then so would consist of both the old and the new one. So not much should change in between this generation and the next generation. Both of them would have DNA molecules, double helices there. They are a hybrid of both N14 and N15, including DNA. Obviously, in this case, this hybrid that wouldn't be right in the middle will start becoming more and more rich on N14. So we'll still be in between these two bands, but a bit closer to N14 and N14. Right? But it will still be not at the level of N14, and obviously not at the level of N15. So it will be something here, this band, at this level. Well, what did they see? What did they see is that they get two bands, not one as you would expect with the dispersive replication model, but two of them which indeed are compatible with a semi-conservative replication model. And by doing that, at this point, they excluded the dispersive replication model, not really proving, but highly supporting. In science, you never prove anything. Your experiments support, they never prove anything. They can always be proven wrong, what you come up with. But highly supported a semi-conservative replication model that of DNA replication. Let's move on to the next topic now, still part of DNA replication. How is DNA replication done? You have done this in Bio 2. We're still going to review all the steps, and I'm going to add quite a bit of additional information in terms of the structure of DNA polymers, the main actors on this process, and the differences between eukaryotes and prokaryotes. So, DNA polymers. There are several DNA polymerases involved in replicating DNA. In Bio 2, you learn about the prokaryotic DNA polymerase 3 and polymerase 1. But in reality, there are other polymerases involved there. Now, no matter which DNA polymers you talk about, they all work when it comes at least to polymerizing new DNA strands, they work in a similar way. They all work in a 5' to 3' direction. What does it mean practically speaking? What it means is that they get the DNA to grow, the new DNA strand to grow by adding a new nucleotide to the hydroxy group in the 3' end of the newly growing strand. It's very important here. So what it means is that they can only attach this nucleotide to this hydroxy group and not to the phosphate group. It's a completely different reaction, right? So by doing that, if you attach the new nucleotide here and then the next one to here and so on, you can see how the new strand is growing from the 5' direction towards the 3' direction. Hence, we say that they work in the 5' to 3' direction. Okay. Another important thing for DNA polymerases, differently from RNA polymerases, which we'll talk about later on when we talk about transcription, DNA polymerases, they need a RNA primer in order to work. We'll talk about that when we talk about DNA replication, the steps of DNA replication. Now, another important information here is that DNA polymerase doesn't really work on its own. In reality, it's kept attached to the DNA by the help of another protein, which is called a sliding DNA clamp. This is the three-dimensional or tertiary structure of a sliding DNA clamp here. This DNA clamp in reality works as a dimer as well. You have two of them. I'll show you in the next picture. But this is what is bound to DNA polymerase. It binds to the DNA and therefore keep the DNA polymerase attached to the DNA. Therefore, allow DNA polymerase to polymerize the new strand of DNA. Where is the new strand of DNA polymerized is within the active site of DNA polymerase. DNA polymers in their tertiary structure, they look like a hand. Basically, imagine a hand, and then you have the double helix or the open double helix here inside the hand right here. The active site is deep inside this open hand structure here, and that's where the new nucleotide complementary to the template strand is brought in and attached to the hydroxy group on the 3' of the deoxyribonucleotide, last deoxyribonucleotide in the 3' end of the growing strand. Okay. Now, what are the steps of DNA replication? Let's get into more familiar territories with you based on Bio2. Well, the steps can be divided into 3 parts. This is just a convenient way to divide DNA replication. The same thing for RNA transcription. Is just an easy way to get you to memorize the steps. In reality, these steps are all overlapping. Okay, it's not that first you have unwinding and then there's elongation and then everything stops, termination. No, they overlap. Okay, so what are the 3 steps? You can also find them named in a different ways. Unwinding sometimes is called as initiation. But so first we have the unwinding of the DNA. In order for the DNA polymerase to access the template strands, the DNA has to be open. Okay, that's the first step. We'll talk about that in the next slide. After that, DNA polymer can start adding the nucleotides. Okay, it needs a help from RNA polymers called primase. We'll talk about that, and this is called elongation. And then these RNA leftover molecules, due to the product of primase, are removed in the last step, which is referred to as termination, where the RNA is removed by a different DNA polymerase and replaced with DNA. So let's start with DNA unwinding. First step. This is a step that takes place at regions in the genome called origin of replications. Now, the big difference already between prokaryotes and eukaryotes is that prokaryotes, they have only one of them. Whereas eukaryotes, because of the huge size of their genome, which is at least 1,000 times larger than the one in prokaryotes, and also because of the different numbers of chromosomes compared to the single chromosome found in prokaryotes, now they need multiple origin replication. But they all work in the same way. So they are all adenine and thymine rich regions. Okay. The reason why they're rich in adenine and thymine is because these two nitrogenous bases when they're found opposite to each other in the two complementary strands, they form only two hydrogen bonds. Practically speaking, means that now the energy to separate two DNA strands reaching adenine and thymine, the energy required to do that would be less than if you had a lot of cytosine and guanine, which have carried three hydrogen bonds for each pairing. So these regions are rich in adenine and thymine, and the opening up doesn't happen spontaneously, but it's helped by this enzyme called DNA helicase. This is the enzyme that opens up the origin of replication. Now, DNA would tend to go back, okay, and rewind, right, reform the double helix. This doesn't happen because of the help of some proteins that I'll talk about in the next slide. But one important thing is that once DNA opens up, now you start having that replication will go, DNA replication will go in both directions, following what we call the two replication forks. So you have two opening here, one on the left side here, one on the right side, which are referred to as the replication forks. When we describe DNA replication, we focus only on one replication fork, but the same thing applies to the other one. The only difference you see when we talk about lagging and leading strand is that they're opposite to each other depending on the replication fork you're talking about. But as we say before, helicases open up the DNA at the origin replication. DNA would tend to reform the double helix. However, this is prevented by the binding of the single-strand DNA molecules by this protein called single-stranded DNA binding proteins, or SBPS. Please use the full name if I ask you about that on a test. Now, they bind it, and by doing that, they prevent the reassociation again of the two strands, forming again the double helix. So DNA stay open about them. However, there is still a problem, is that while this separation keep going and going and on, it would be causing a tension here ahead of the replication fork. So if you look at this replication fork here and you keep opening up the DNA in this direction, now you start having tension, more and more tension building in between the two DNA strands. So it's like if you take, imagine you take two rope, you tie them on one end, right? Then they're twisted around each other. And now, so one end is attached, let's say, to a wall. You take the two ends at the opposite side and you keep pulling them apart. You see, if you think of, you start having more and more resistance there. Now, usually a rope would not break. They're stronger probably than you are. But when it comes to DNA, this tension will lead to the breakage of these DNA strands. To prevent that, you have this enzyme called topoisomerases, which do cut one of the strands, and by doing that allow that strand to unwind around the other one. Then the DNA gets joined again, and by doing that they release this tension. So in DNA unwinding, DNA is open at the origin of replication by DNA the helicase, single strand binding proteins that prevent the reassociation, and topoisomerase prevents the increase in tension ahead of each replication fork. In the next step, elongation, that's when the DNA gets synthesized. Okay, so the template DNA on one of the two sides of the replication fork get copied into the new strand. This is done by DNA polymers, right? Which, as I said before, they get the new strand to grow in the 5' to 3' orientation. As I was mentioning before, though, too, one of the problems of DNA polymerases is that they cannot start out of nowhere. What practically speaking means is that if you take one template, let's say take a sequence of DNA, okay, a single strand sequence of DNA, add some nucleotides, so deoxyribonucleotides, right, 3-phosphate, adds DNA polymerase would not start copying that template strand. It cannot start from zero, from nothing. Only it can do is specialize to attach a nucleotide to a pre-existing nucleotide. But if there is not a single nucleotide attached to the other template strand, they cannot work. So for this reason, DNA replication or the new strand has to be started by another enzyme, which is an RNA polymerase called RNA primase or primase. These polymerase, unlike DNA polymers, can actually start attaching nucleotide to each other in a complementary way compared to the template strand, even if there is nothing in there. However, it's not working with deoxyribonucleotides, but it's working with ribonucleotides. It's an RNA polymerase. And so what it does, it produces these short RNA primers. You see them here in blue. It will be like 10 nucleotides long. And by doing that, so it can start from nowhere, here and there. And then by doing that now, at this point, it prepares a 3' hydroxy group for DNA polymers to start working from. When it comes to prokaryotic, and that's what we are really presenting here as the prokaryotic, the simplest DNA replication, the enzyme now that start attaching the deoxyribonucleotide now from this 3' is DNA polymerase 3. So basically what it's doing is extending this RNA primer here by adding one nucleotide complementary to the template strand, one at a time. So it will keep going, and going, thanks to the sliding clamp here that keeps it attached, going always in the 5' to 3' direction. By doing that though, it's not producing a pure DNA strand, but it's producing RNA DNA hybrid molecules. So this being RNA and this DNA. Now, in each replication fork, now you can see that because of the way it works with the RNA primer and the inability of DNA polymers to go 3' to 5', but being able to synthesize DNA only 5' to the 3' direction, you can see how only one of the two template strands, so you would have for each fork one template strand here and one here. Now, once the RNA primer is synthesized, only one of them can be copied continuously. The strand that is copied continuously is referred to as the leading strand. Now, and the other one, so if you have an RNA primer here, DNA polymers can only go 5' to 3' in this direction. So what it means is that the more the DNA open, the more you would have a gap that cannot be replicated. So what it means, practically speaking, is that you need that in this strand, which is referred to in this case as the lagging strand, you need RNA primers to keep synthesizing always new RNA primers. Okay. Now, keep in mind one thing. This is the lagging strand and this is the leading strand for this replication fork. But if you think now the other replication fork, which we are not seeing here, right, on the left side, it will be the two strands are the opposite. So the definition of leading and lagging strand is not a definitive definition for each strand. A leading strand on one fork is the lagging strand on the other one. Okay, and vice versa. Now, these generate these short fragments here that are referred to, so RNA and DNA molecules here, which are referred to as the Okazaki fragments, okay, all joined covalently. Now, this is a picture where I want to show you a more realistic way of seeing how the DNA polymerase actually is working through this. Now, it looks like DNA polymerase is going in one direction here in this strand and on the opposite direction here. But in reality, DNA polymerase works as a dimer. So two units of DNA polymerase work together on both strands at the same time, going in the same direction. How? By folding one of the two strands. And that's exactly what's happening here. So this will be the leading strand, all right, here in red, purple, whatever color you see here. And this in darker red will be the lagging strand. But you can see how by folding this, now you have two sliding clamp and two DNA polymerase 3 attached to each other. Now this allowed for the DNA polymers, actually both of them going in the same direction, even though it looks like they're going in opposite direction when we present it in this way. And now you have here one type of topoisomerase DNA gyrase here, which is actually releasing the stress ahead of the replication fork. So this is what happens up to, forget about that part there, elongation. And then at the end, but in reality this is overlapping with elongation, this phase here. The third phase is referred to as termination. So in the end, once you have that the two replication forks move, imagine you're talking about a circular prokaryotic chromosome, right? The two replication forks are moving away from each other until they will meet at the opposite end of the circle, right? At that point, you would have had that both strands have been copied into a complementary sequence. The problem you have, though, is that that is not a final complementary sequence. The reason being that you still have these RNA fragments within the newly synthesized strand, right? But you're doing DNA replication, not hybrid DNA RNA replication. And so these RNA primers, they have to be removed before DNA replication ends. How is it done? This is done by a different DNA polymerase, DNA polymerase 1. And again, I stress we're talking about prokaryotic DNA polymerization right now. DNA polymerase 1, what it does, it has a different property compared to DNA polymerase 3, which is the ability to actually remove nucleotides. So it has the same capacity of synthesizing a new RNA strand in the 5' to 3' direction. However, at the same time, it has also the ability to go in the opposite direction, 3' to 5', to remove nucleotides. In this case, to remove ribonucleotides. The 3' to 5' activity is referred to as the 3' exonuclease activity of DNA polymerase 1. Exonuclease means is removing nucleotides from the ending. So it's starting from the ending of the RNA primer, going 3' to 5', removing them, and then it synthesizes the new DNA going back to work in the 5' of the 3' activity. In this point, so the final result being that basically the RNA or the ribonucleotides in the RNA primer at this point will be replaced by the corresponding deoxyribonucleotide in the sequence. Therefore, by doing that, the RNA primer are replaced with DNA sequences. However, this is not the last step. DNA, you're still left with a little gap, one phosphodiester bond missing here. In between, you can see in this picture right here, in between now the DNA strands here. DNA polymerase 1 cannot form this last phosphodiester bond because these actually will be going in the 3' to the 5' direction. It cannot do that. And so this is done by the final enzyme that I'll talk about here, which is DNA ligase, which all it does, binds to this area where this phosphodiester bond is missing and it forms it. And by doing that, now you would end up having that all the RNA primers are being removed. Don't see it really in this picture, but all the RNA primers will be being replaced with DNA sequences at this point, and all the fragments would have been joined together by DNA ligase. At this point, you would have finally have a two identical copy of DNA, both double-stranded and made only by deoxyribonucleotides. This is just a summary of all the enzymes that we talked about here. You know pretty much all of them. Probably the one I might have introduced new here is the sliding clamp. I'm not sure if you talked about that in Bio 2, but this is that protein is not really an enzyme, but it's a protein that binds to the bind to the DNA, and by doing that binds also to the DNA polymerase, and by doing that it keeps the DNA polymerase attached to the DNA so that the DNA polymerase can do its polymerase activity. Some differences here now between prokaryotic and eukaryotic replication. There are several differences. Okay, again, the process that I described refers basically to the prokaryotic DNA replication. The steps are very similar in prokaryotic and eukaryotic replication, but there are some differences. First of all, the starting of the whole process is a little bit more complicated in eukaryotic cells. The reason being that DNA is wrapped around histons, right? It's not just made by ribonucleotide, but the double helix is wrapped around forming nucleosomes, do you remember, around histones and so on. So first of all, the DNA has to be unwrapped in eukaryotes, something that doesn't happen in prokaryotes. There are no histons there. Second thing, the process is lower in eukaryotes compared to prokaryotes, about 10 times lower. If you get something like 500 to 1,000 base pairs per second in a bacterium like Escherichia coli or E. coli, you get something like 50 to 100 bases per second in a eukaryotic cells, so roughly 10 times lower. Other thing, eukaryotes, they have multiple chromosomes, longer chromosomes, so they don't rely on a single origin of replication, but they have multiple of them. I already mentioned that when we talk about the unwinding of the DNA. Also, there are many more DNA polymers in eukaryotes compared to prokaryotes. I have a couple of slides coming up about that, and also that the way that DNA methylation is done. So these are the polymers, or the main polymers. There are many polymers in E. coli, but in general there are less than the one that there are in eukaryotes. So these are DNA polymers you find in there. Now, the really important one for DNA replication or synthesis of new DNA, as is DNA polymerase 1, which is the one that removes the RNA primers, as we said before, replacing it with new DNA, and DNA polymerase 3, which is the one that extends the RNA primers. Then DNA polymerase 2 is involved in DNA repair. If there is any mistake introduced by some mutation and so on, you would have DNA polymerase 2 that does that. And then you have also DNA polymerase 4 and 5, which takes place in really serious damage to the DNA. But the key one I want you to remember is DNA polymerase 1 and polymerase 3 here. When it comes to eukaryotes, you have a huge amount. I didn't even write them all here. Okay. Now you have that, but the important ones here, you can recognize some of them. The important one is the alpha, which is the one that starts the whole process here. Now you have also the lambda and epsilon one are extremely important. So all of these three are involved ultimately in the DNA replication, right away the process that we described before. So you have multiple versions there involved in the process there. Now you have the beta one that is involved in actually do the DNA replication in the mitochondria particularly, and also acts as a repair enzyme as well. The lambda as well, it's involved in mitochondrial DNA synthesis. And then you have so many other ones there, right? Lambda, sigma, and so on, micro, and so on. And here you recognize telomerase, which is the one that is responsible of doing, if you want, the tweaking of DNA replication at the end of the chromosomes, basically preventing them from getting shorter and shorter over time. And this enzyme, though, it's active not in all cells, but only in stem cells and germ cells. So the cells that are involved in the regeneration of tissues or the production of new organism, right, in the replication, in the fertilization process. Okay. Now, another difference that I mentioned before between prokaryotes and eukaryotes is what happened to the DNA after the synthesis. So oftentimes DNA doesn't exist only as sequence of nucleic acid as it is, but they get modified. And a big modification that takes place is methylation. Methylation plays a key role in different things. First of all, is one of the epigenetic mechanisms used to regulate the activity of the genome. Highly methylated areas in eukaryotes, for example, are inactive. They are not transcribed into a protein. Even though the gene is still there, does not get activated because of methylation. So methyl groups can be added or removed, the inactivating or activating the specific gene. There are other epigenetic mechanisms other than this one, but here we focus on methylation. When it comes to prokaryotes, methylation is involved in the prevention of the activity of the same enzyme that some bacteria produce, restriction enzyme. We'll talk about that in the lab. Restriction enzymes are enzymes that cut the DNA as specific sequences. They literally cut both strands that separate the DNA. And so they break two phosphodiester bonds in both strands, okay, of the DNA. And now they are produced by bacteria. But bacteria have their own DNA. Obviously, you don't want these restriction enzyme to act on the bacteria genome itself. It's just used as a mechanism to prevent infection from viruses and destroy the viral DNA. And how is prevented that? Through methylation. Now, the methylation, though, that takes place in prokaryotes involve adenosine, so which is methylated right here. Sorry, there's a mistake here. This is not present here, so Don't look at this. It's not here. Methylated is the methyl group is attached here right now. There's a mistake there. And whereas in eukaryotes, methylation takes place in cytosine, not in adenosine here, and takes place here on this carbon right here, part of the ring. Again, add it and remove it over time. I'm gonna stop here for the first part. In the second part, we'll talk about transcription, all the steps, and then we'll finish up all the way talking about how replication takes place in RNA viruses.